Hey guys, so in this video we are going to be doing the second section of chapter 20 when we are talking about antibody and antigen complexes. So with these guys, we mentioned before that antibodies are going to attach onto things and bring them together. So if we were talking about them bringing cells together, like in the red blood cell test, when we're testing about what blood cell type you have, are you A blood type, B blood type, that was agglutination because those were bringing the two cells together and they were cells. In this case, we're talking about the antibodies are attaching and bringing together the antigens. So when it's antigens instead of living cells, it is precipitate. So this is precipitin. They are our antigen antibody complexes. Antigen antibody complexes. And these things are going to bring together multiple different antigens on there. And remember, we talked about how antigens could have different epitopes. They could have 3 different ones on our example that we had before of the different epitope types. So we also talked about polyclonal and monoclonal antibody serum too. And poly was all the epitopes that would work with our antibodies. And then monoclonal was one where we were very, very specific. We went through extra steps to make sure that we only got the one antibody that interacts with the one epitope that we want to deal with. So monoclonals were just the one, while polyclonals were multiple epitopes. So you have a better opportunity, a better chance of attaching onto the antigen with the polyclonal antiserum. So that's the stuff that we're going to use when we're talking about these tests coming up. We're going to talk all these different types of tests. And again, remember we talked about how things kind of affect this bond. And so the higher affinity, the stronger the bond is between the antibody and those antigens. And if it was a weaker affinity, you might have cross reactants going on. So if you put that antibody in a serum with different antigens that are kind of sort of the same structure, then it might attach onto them and cross react. So higher affinity, the stronger the bond, the less cross reactivity we need to worry about. So we said that they are going to form antigen antibody complexes. When they do this, we are going to try and find this optimal place. This optimal place is called the equivalence zone. So when we are in this level, we have more antibodies in the serum than we have antigens. There's only 1, 2, 3 antigens here in this, and we have bukus of antibodies in this serum. So this is a zone of antibody excess, too many excessive amounts of antibodies. Over here, we have a zone of antigen excess because we have 1, 2, 3, 4 antibodies, and we have bukus of antigens. So we want to find this nice place where it's a 1-to-1 ratio. That is the equivalence zone. So where we have equivalence zone, we have enough antibodies to attach onto the antigens. They're a perfect amount, and they will form this perfect lattice work. That is what we're looking for. The equivalence zone is where the antibodies and antibody complex are going to come together. We're going to have perfect precipitin showing up when this happens in the equivalence zone, where our ratio is roughly the same for antibodies and antigens. How do we know what that magic level is? That's what we're going to test for. We're going to try and find that zone of equivalency. So with our precipitin ring test, the way it works on precipitin ring test, you're going to have all these different test tubes. And in the bottom of the test tubes, they are going to have this antigen solution. So all of these have this antigen solution in there. Then you are going to have another substance at the top, and that is going to be your antiserum. Your antiserum will vary. While your antigens are all the same, your antiserum is going to undergo a serial dilution, which we have talked about before, where you have a concentrated substance and then you pull a little bit out and you dilute it. And then you pull a little bit out of that and you dilute that. A little bit out of that one and you dilute on and on and on. So we have it as, 1, and now we've got a 1 to 2. So we're half as concentrated in this level than we were before. 1,4, 1,8, 1,16, 1/32. So This part, the top, the serial dilution antiserum, varies in the different tubes. That's what's going to set the tubes apart. And then we're going to add in glycerol. When we put glycerol into that antigen solution, it keeps the antigen solution separate from the antiserum to where they only interact in the middle, where both of those substances are going to touch. So when they interact in the middle, remember how we said we could have antibody excess and we could also have antigen excess. But when we were right smack in that happy place of a 1-to-1 roughly ratio, then that was the zone of equivalence. That's what we're looking for. We are looking for precipitin to happen. We want to see precipitation. We want to see that precipitin ring happening. Then that's going to tell us that we have that happy ratio going on. The zone of equivalence is going on. So in our tubes, we have it at 1. I'm not seeing anything here. There's nothing here. At 1/2, there's nothing going on. But at 1/4, at 1/4, we start to see precipitin rings. That glycerol that's separating these things out, again, remember, that's where we're going to look between the two different levels. And are we seeing precipitin antibody antigen complexes happening? And we do. They are going to form these precipitin rings happening. So what we're looking for is we are going to find the least concentrated out of our serial dilutions. Who's the weakest serial dilution where we can still find a precipitin ring? So in this case, we found a precipitin ring here, and we did not have one here. We missed our zone of equivalency. It's not equivalent anymore. We're not seeing a ring anymore at 132, so that's not useful. We need to focus on the ones with the precipitin rings. So there was 1/4, there was 1/8, and there was 1/16. 1/16 is the magic one where we still find a precipitin ring. So we're going to use that, and 1/16 is the highest dilution where we still see a precipitin observed. So that's our titer. At 1/16, we're going to take the reciprocal, which is 16, the bottom number. The reciprocal is the bottom number. We're going to take that, and that's our titer number. So for this example, titer is 16, where we still see a precipitin ring with the precipitin ring test. So this whole concept of this zone of equivalence, where we have the proper balance between antigens and antibodies, and we form precipitin, we form this lattice work, that's going to be our red flag. That's what we're looking for all of these tests. So we found it in the precipitin ring test. There were 3 different serial dilutions, and we go for the lowest one, which was 1/16. And so our titer is 16 in this example. That's the precipitin ring test. There's another test called the Ochterlony test. I think it looks like Ouchterlony, so I call it the Ouch test. But however you want to pronounce it, I'm sure I'm slaughtering it, and I'm sorry if you know the proper pronunciation, because I don't. It may be Ochterlony. But anyway, what we're looking with this test is basically the same principle going on. So what is this test doing? What's going on in the Ochterlony test, the Ouch test? We have this central place here. So we're going to have our gel, our agarose, our gel dish, and in the middle we're gonna carve out wells. We're gonna carve out wells, and we have what, 5, 1, 2, 3, 4, 5 wells around them, and then in the heart of the center of this thing, in the middle, we are going to put the antigens. So here's our antigens, and the antigens are going to diffuse through the gel. So the longer it sits there, the longer the antigens are going to diffuse through. Now, out of all of these things, remember we had 5 things in these wells over here. These are going to be different antisera. And again, antibodies vary. They have to be very, very specific. So they're only going to match up with that one thing. Hopefully, as long as they're not cross-reactive, they should only match up with the one thing that you're hoping that they match with. So this would be a good test to see if there's any cross-reactivity. And in this case, only one of them is interacting. And how do we know that it is interacting? Because we see the precipitin arc. So number one right here, this is our star student. Number one is interacting with the antigens in A because it forms this arc here that shows up. And what is the arc? It is precipitin, where we have that proper balance, that equal, even Stevens, the zone of equivalency between antigens and antibodies. And it only happened with number one. So this is the double immunodiffusion or the Octoloni assay test. So that one can help us figure out which anti-sera works for us. Is there any cross-reactivity? That would show in that test. The next test, the RID, the RID assay, or the radial immunodiffusion assay. For this one, we are not looking necessarily to see which antiserum works. We're wondering what the concentration level is, because we have a mystery, unknown concentration, and we're trying to figure out what it is. So in this case, we are going to have antigens, and the antigens will be in various concentrations. So as we go up from 1 to 2 to 4, 1 to 2 to 3 to 4, because I can count, our concentration of the antigen goes up. So we are going higher and higher in our concentration of the antigen as we are creating these examples, 1, 2, 3, and 4, to compare to. So these are our stock examples that we are looking to for different comparisons. And these guys will diffuse across the agar and form a zone of precipitation. And we want to compare these known zones of precipitation to an unknown. So here's our unknown. Number 5, we don't know what it is, but we're going to treat it the same as the others. We're going to pour this agarose, put this unknown in here, and then watch the antigens and antibodies interact. And then we will compare it. So we're going to measure this zone of precipitin, and try and figure out which concentration level it's at. And so when we look at its zone versus the other zones, it falls somewhere in between 2 and 3. So it's somewhere in this zone right here on its concentration level of the antigens. That's what we're trying to figure out with our radial, because it's circular, immunodiffusion RID assay. So very helpful to determine concentration levels. Another type of assay that's not pictured, but it mentions it, is the flocculation assay. And with the flocculation, it's similar to our antigen antibody complexes, but it is using lipids instead. And so the lipids, when they react, they're going to have this kind of frothy foam that you're looking for. Instead of a precipitin ring, you're looking for foam that's going to be on the test tube. That's a flocculation assay. Neutralization assay. So antibodies did all sorts of things, right? They cause agglutination, they can cause precipitin, they can cause opsonization, which basically is like a giant neon sign flashing for the nearby phagocytes to come along and eat it. And then another thing antibodies do is neutralize stuff. So with a neutralization assay, we have all these plates in this picture. So we have 6 here, and we have 6 here, and we have 6 here. And so the way it works, they are going to have, all of these started off the same. They all started off the same, and they are all going to have this layer of cells, living, cells in the agarose on the bottom. And then they are going to put the viruses on top in another thin layer on top of those. And as the viruses diffuse through the gel, they are going to do what viruses do. And what do viruses do? They attack cells, right? So those viruses will get to the cells, and if we remember our lytic and lysogenic life cycles, the cells are going to take in the virus unknowingly, brings it inside, and it reproduces, hijacks the cell, and then ends up killing the cell, lyses the cell. So when that happens, you're going to see, instead of these pretty purple, everything's uniform, when it kills the cell off, it's called a plaque. It's a plaque. And so over here, the virus has taken out pretty much all of it. It's just coated in plaques, and it's so clear because of that cytopathic killer effect that the virus does. On the other end, over here, we have a serum sample with our antibodies, our virus-specific antibodies. And so if we have virus-specific antibodies, one of the things antibodies do is neutralize the virus. The virus can't get in your cell if you have covered the virus in antibodies, and it cannot attach on to the cell, or you have blocked the attachment points on the cells, so the cells can't bring in the virus, and the virus is just left out hanging with nowhere to attach. So with the neutralization assay, that's what we're putting in over here. And so we have very high concentrations over here. None of those viruses got in here. Everything is still pretty in purple, and those cells are all still alive. We don't see any plaques on this end, while this end is like 100% plaques. It's all clear. All those cells died off. They all got infected with viruses. And over here, we have 0 plaques because we've neutralized everything. And what we're looking for is what concentration is going to give us about a 50% reduction. We are looking for a 50% reduction in the plaques. So where it's about 50% purple and 50% plaques are showing, that's what we're looking for. So these guys right around here where they've got the Sharpie is sort of where we're looking at, somewhere around there. Okay, where's the hell? Some purple healthy cells still around, but some of them did get infected with the virus. That's what we're looking for. We're looking for a 50% reduction in the plaques when we use this. And that's going to be our titer. So whatever that concentration was, where we see 50% of the plaques disappeared, we have 50% of the plaques are there, 50% of them are gone, and the living cells are still alive. That would be whatever that concentration is, that's our titer. That's what we're looking for. So I think the textbook had mentioned, they just popped out some numbers, and they were like, in this instance, this solution was at 64, so that's our titer. Our titer is 64. But in a neutralization assay, whatever that concentration was, we're looking for the 50% plaque mark. Okay, so we are going to shift to electrophoresis. And remember, we talked about electrophoresis, I think, in chapter 12. And hopefully you got to run one. If you had Bio 1010 or Majors Bio 1107 before you came to your AP1, AP2, Med Micro bios, you got to run one of these, at least if you did it at Georgia Highlands, you got to run one. I hope you did, because they're really cool. And we mentioned them in chapter 12. And with the gel electrophoresis, we can run DNA, we can run RNA, and remember, when we ran a gel, that these things are going to follow the current. We said the DNA was negatively charged, so it's going to go towards the positive side. And the longer you leave it, the longer those little pieces can move across. And then you will stop it as the pieces, get near the end. The smaller pieces, they move the fastest. The bigger pieces got stuck behind in the gel. So it goes from biggest pieces down to the smallest pieces because they move the fastest when you're looking at an electrophoresis. Again, and it can be DNA, it can be RNA, it can be proteins. What do you mean proteins? It can totally be proteins. So the first person who was doing this was, I'm going to slaughter this name, RNA Tiselius. And this was back in 1937. And this person ran the first protein electrophoresis. So we looked at DNA and we looked at RNA, but we didn't even know about DNA and RNA because we didn't even know their structures, what they look like, until the 50s. So before this, proteins, because we still weren't sure, we talked about how we weren't sure where the genetic material was coming from, and was it proteins, was it DNA, what was it? So in the 30s, Tiselius produced the first protein electrophoresis. And if you remember, proteins are made out of amino acids. So there was the amine head, the carboxyl group, and then the remainder. And the remainder was about varied in the 20 different amino acids. And some remainders were charged as a positive charge. Some of them had a negative charge. Some of them are non-polar. Some of them were polar. They varied, and that's how we categorize the different amino acids. So we can run an electrophoresis when we know the charge of these things and split these apart. So when you run a protein electrophoresis, the smallest protein is albumin. When you're going to run blood, remember we talked about blood has lots of things in the plasma. It's proteins, these little guys floating in the plasma, and there are immunoglobulins and there are albumins. Albumins are small, and the immunoglobulins or antibodies are a little bit bigger. So we have antibodies here, and they were first named, with the Greek alphabet. So we had the albumins were here, and then we went alpha, beta, gamma, and the alpha was actually 2. So we have alpha 1 and we have alpha 2. So in one of these, you can see these, yeah. So there we'd have alpha 1, this looks like alpha 2, then we got beta, and then gamma up here for your different antibodies. So you can run an immunoelectrophoresis if your patient has abnormal, it has proteins in the urine. We shouldn't see proteins in the urine. So let's test it and see what, what's running through and compare it. So here we have run a page. We ran a polyacrylamide gel electrophoresis. And with the page, we are going to compare, and we have normal serum, patient's urine, and we compare that throughout. Remember we said that the proteins will spread out on how small or big they are. So here we have our different ones, IgG, IgA, and these two here to compare normal versus patients, normal versus patients. So we run the page, and then we're going to put these anti-sera in the troughs here, in this guy, in this guy, in this guy, and we are going to see precipitin. I mean, they're not necessarily rings. They're more arches. So precipitin arches we're going to find. So you can see them. There's one here and here, and we can see where that's happening. Does it match up? Is it normal or abnormal with this immunoelectrophoresis IEP test to measure proteins? What's going on with the proteins? Speaking of proteins, we can run proteins through a gel electrophoresis too. So the previous one with the immunoelectrophoresis, we did a page first, and then we put anti-sera in there in these little troughs, and then we looked for arches. With a Western blot, we are going to run it. It's just like the Northern blot and the Southern blot with our DNA and our RNA. We are going to run it through. So we're going to run it through, we're going to separate out the protein. So we're going to run an electrophoresis on the gel, and then we're going to blot. So just like it sounds, like you would take, a paper towel or a napkin, and you are going to transfer stuff. So when we run the gel, here's our gel sheet that we get left over, your very delicate gel, and then you're going to put this membrane next to it because you want to transfer the proteins from the gel onto the membrane sheet, because the membrane sheet is what you're going to use from here on out. How do we get the protein stuck in the gel, out of the gel and into the membranes? Well, electricity, just like how we got the stuff to run through the gel. So we're going to have the gel sandwiched with the membrane in between a filter paper and a filter paper. And we make the filter paper sandwich, and then we have electricity. We got the negative charge over here and the positive charge over here, and that is going to pull the proteins out of the gel and into the membrane. And it depends on what proteins you're looking at. If it is hydrophobic, then it's a different type. It's not nitrocellulose. It's going to be a PVDF membrane. But regardless, you're going to pull them from the gel into the membrane. Now, we have our membrane. What do we do with it? Here is our membrane with our transferred proteins over here. we're going to have a series of steps. We are going to look for the antigen that's stuck on that membrane, and then we're going to find an antibody that goes with that. So the first one that attaches onto that antigen is called the primary antibody. Makes total sense, right? So we have the antigen, the antibodies are attaching on. Then we are going to have a secondary antibody that attaches on to the first one. The secondary antibody, because we're looking for this one, that's the one that we're looking for, that one protein that's got the antigen on it. The secondary antibody is going to have something that is going to be our signaler, our trigger, something that's going to let us know what we're looking for is present or not present. So it's going to have an enzyme on there and something like a fluorophore or a chromatophore, something that is normally clear, but if you get the reaction you're looking for with the substrate, then it will change color or it will fluoresce under light or something like that. We need it to, give us a signal. This is the signaling molecule. So we had our primary antibody. Then we go to our secondary antibody, and that's going to have our enzyme attached to it with some sort of signal molecule, again, fluorophore, chromatophore, whatever it is. And then we're going to add the substrate. Did we see a change? Did it fluoresce under the light? Did it change color after we added the substrate? If it did, then that's what we're looking for. And if it didn't, then it didn't have it. And these things, if it didn't, the antibodies didn't attach. You didn't have that protein that you were looking for. So this is a way to pull the proteins out and then see if we can identify a very specific protein. And that's the one that the antibodies will stick to. If you have it, the antibodies are going to stick. If you don't have it, the antibodies won't stick. So this is Western blot. If you get the protein that you're looking for, you're going to see a change. So, it will fluoresce or it'll change color. If you don't, then you didn't have the protein on there that you were looking for. Examples of protein tests. This is an HIV test in the bottom. And so we have our negative strip and a positive strip. These, I didn't get a whole lot of explanation in the textbook, so I, consulted Dr. Google. And as far as I can tell, and I may be totally wrong, as far as I can tell, these are HIV-1 proteins in this positive control. The negative control is missing those. The negative control does not have the HIV-1 proteins, and it does not have the HIV-2 proteins. This, I believe, is HIV-2. Okay, so these are our controls. Negative is not going to show the proteins, while the positive does show the proteins, both the HIV-1s and the HIV-2s. So let's look at the patient and see what's going on with this patient. And if I'm interpreting this correctly, it's not my wheelhouse, but I think this is what's going on with here. I think this patient sample is telling us that this patient is positive for HIV-1 antibodies, that they have been exposed, they have HIV-1 antibodies. They have not progressed to HIV-2 though, because you don't see, this is the HIV-2 protein, and you don't see it. It's missing here, it's not there on these patient samples. So this one, HIV-1 positive, but not for HIV-2. And this one, I'm thinking does not have it, but there's this strange, I'm not quite sure what's up with that one. But I'm thinking they're probably negative because they don't have the whole gamut of them. Again, I may be wrong, but that's how I'm interpreting this sample for this. What I really want you to know is what a Western blot is. What is your basic takeaway from this? Know what, Northern, Southern, and Western electrophoresis are running. Are we looking at DNA? Are we looking at RNA? Are we looking at proteins? And kind of have a grasp on what a blot is, that we are going to pull it from the gel into a membrane, and we are looking for specific things. If we have it, those antibodies will stick, and we will have a color change or a light change or something like that. If we don't have that present, the protein that we were looking for, those antibodies don't stick. They will wash away, and you're never going to see that protein light up. like you want to because you're missing it. The antibodies don't have anywhere to stick to. You're missing your protein, you're missing your antigen to attach to. So this is Western blot. Complement fixation test. So this one is a comparison to see, does this patient, we are looking to determine whether a patient's serum has specific antibodies. Do they contain antibodies to a specific antigen? So let's see, how do we test? Does this patient have antibodies or not? Let's see, we got two patients in this example. So here's patient A, here's patient B. Patient A has pretty green antibodies in there, and both patients have these pretty purple complements in there. And if we remember the job of the complement system, when we learned about this in AP2, that the complement system had a classical or an alternative pathway. Classic is where you have the antibody attach on to the antigen, then the complement system attaches on to the antibody, and then eventually you are going to end up with a cascade that cell ruptures and dies, and we kill it off with the complement proteins. That's their job, and everybody has complement proteins. So in this case, patient A and patient B both have complement proteins in there. They both have purple complements, but only patient A has antibodies, and that's going to be important later, because in step 2, we're going to heat this stuff up. What happens when you heat up proteins? Do they still work? Nope, they don't. They denature, because we said protein structure was key to proteins. So if you heat them up, they're gone. Poof, gone. So now all we're left with is patient A and their antibodies, and there's nothing in the patient B serum now. Now we're going to add stuff in and see what happens. So we are going to put equal amounts of complements back into the system. So purple returns, the purple is coming in. And then antigens are added as well. So with patient B, we've got purple complements floating around in there and little orange antigens floating around in there, and they're all kind of separate doing their own little thing. In patient A, we have complements, but they are attached to the antibodies, which are attached to the antigens. So we have a whole complex going on, which we said is what happens in real life. You got the antigen, antibody attaches onto that, complement's going to attach on the antibody, and then boom, eventually you're going to kill that cell. So that's what we're starting to see here. Patient A has antibodies attached to the antigen and the complement. Patient B, everything's floating around because they don't have the antibodies to unite the other two pieces. So now in step 4, we're going to add some red blood cells and some antibodies against the red blood cells. So we have sheep red blood cells and antibodies against the sheep. Well, with patient A, we already said that the complement system was activated. We had the antigen and the antibody and the complement all attached together already. So these antibodies are going to, those anti red blood cell antibodies are attaching on to the antigens of the red blood cells, because that's what they do. But they can't activate the complement system. Why? Because the complement system was already busy with the previous antibodies that the patient always had since day 1. So the little green antibodies are taking up the complement system in, what was this, step 3. Step 3, those complements stuck to patient A's antibodies. So when we added in other antibodies in the red blood cells, they hooked up, but they can't die. We can't activate the complement system because the complement system is busy at the moment. Patient B, their stuff was floating all around because we didn't have the green antibodies floating in patient B. And so when we added the sheep's red blood cells and the antibodies to the sheep's red blood cells, the complement's still free roaming in there. It's not hooked up. So what's going to happen is we're going to have a complement cascade happen. That's what's going to happen. The job. So in patient B, we are going to have the antibodies to the red blood cell attach onto the cells, but instead of just dropping down and doing nothing, complement attaches on, and then we are going to end up rupturing the cell, which is the job of the complement system. So when we rupture the red blood cells, the water, they're going to lyse and the water's going to turn pink. So that's our giveaway. Pink water from the ruptured red blood cells, pink fluid, is going to be our giveaway that patient B didn't have antibodies previously that we were looking for. They didn't have the specific antibodies we were looking for. They didn't hook up and activate the complement and keep complement busy. Complement worked in the end and ruptured the red blood cells. So these did not have the antibodies in patient B, and we know it because the red blood cells burst. Patient A does have antibodies. And how do we know that? We know that because there's no lysing of the red blood cells because the complements were fixation. They were stuck where they are. So patient A, the red blood cells are there, but they're not burst. They're not bits and pieces of them all over the place. Solution is clear. So this is complement fixation. They are fixated if the patient has the antibodies against the to activate and can keep the complement fixated. If the patient does not have the antibodies, then the complement does what complement does, and it will end up lysing the red blood cells that you add in at step number 4. So here we can see different terms all in a table because OpenStax really likes to put all the information in one place, which is cool when you are studying later. All right, so that's the last of this section, and we'll have agglutination assays in a different video.